Well, as you all know, it's a long time that we are interested in TDP-43, that is a protein that has a sort of, is an HNRP essentially, has multiple functions, and the structure is classical of an HNRP. You have it below. Nuclear localization signal from the amino terminus, first RM1 domain, second RM2 domain that contains the nuclear export signal, and the C-terminal glycine-rich region, and a series of interactions that we have already mapped in previous publications. This protein is incredibly well conserved and is exchangeable between Drosophila and man, for example. The Drosophila protein works in human cells, and human protein works in Drosophila. And recognize the same target sequence. So the bit that I am going to talk to you now is the autoregulation of the levels of this important protein that actually are quite surprising. I mean, it has, if you express a transgene in cells, a single copy transgene, and you induce it, within 6-8 hours, practically, the endogenous protein disappears. And you can see here, well, here you see that 24, the bottom band is the endogenous, that is the inductor of the transgene. As soon as you induce the transgene, in 24 hours, practically the endogenous protein disappears, and obviously at 72 even more. This function is dependent on the RNA binding function of TDP-43, and you see that the mutant of just 2 amino acid changes that abolish the RNA binding properties of TDP, and you can see that in this case the endogenous protein does not disappear. This is the protein. At the RNA level, we have an equally dramatic picture. These are the two messengers. You can see the messenger, the gene is this one, and the difference between this one and this one is the use of this polyadenylation site or of this one. I mean, this is ending up here, and this one here. These two messengers, obviously, after the transgene is induced for 24 hours, but in this case 72, you get almost total disappearance of the short one and a more resistant. I mean, this is normally less than a third of this one, but is retained after induction, so it's less downregulated. When there is the mutation in the phenylalanines, there you have that there is no self-regulation. Well, you see that here we have marked an area that is hatched, and that is an area where this is the 3' UTR from all this bit, or all this bit according to the messenger. And this area, the 3' UTR, is within a region that earlier, in collaboration with Untitled, a series of clip sites for TDP were mapped that are not the characteristic site of GU repeats, that is the normal target of the protein, but they are a bit less GU-rich and less specific. So this 3' UTR, at least in this area, this is exon 7, what we call exon. All this, the normal protein ends up here, the coding sequence, and this is just all the 3' UTR. What we found is that we stuck this to GFP, we can reproduce the regulation. So at this point, I am going to focus in this, that is the region that have the clips. And I am calling this exon 7 and exon 8 because this is a silent intron. You see it here, is never normally spliced. is always contained or in this measure or in this one, but in certain conditions you will see that it's spliced. And within this intron there is all this series of sequences in bold that are the original clip sequences. Here is the poly A1 that is within one of these sequences, and here is the intron 7 exon 8 junction. So the poly A1 is within the intron, and the poly A2 is afterwards. The poly A1 is the main polyadenylation site of TDP. The only two that are mainly used in the normal cell is this one and this one. These are never used, but this one is used when there is a phenomena of overexpression of TDP-43. First, we map what were the essential sequences. So all this is the 3' UTR. Stop codon there, which were the essential sequence for the self-regulation. And obviously, if we remove just the hatchet sequence, leave it the intron size and the poly A1, what we call this region, we call it TDP-PR, Tdp Binding Region, very imaginative. And that region, if you remove only that region, there is no more regulation. But not only that, you can leave that region in and remove the splice sites by mutations, and then you don't have also regulation. You can express as much as you want of the transgene, and there will be no protein. And there are others that we are going to see. I mean, the poly A2 doesn't make a big difference if you change this, or you reproduce two. You replace the 2 for 2 poly A1s here, one before and one after the intron. The regulation works all the same. The only one that, again, does not work if you delete the whole area there. And you don't have any more. It's just like the cDNA ending in this poly A2. Here we analyze by PCR. Keep in mind that these messengers that you see by PCR, you never see it in the northern. So the amounts come up because we push the amplification, but really in the cell are minimal. And you can see here the induction makes a switch where this intron is removed and the poly A1 with it, and appears this species that is very transient, and we'll see a little bit more about it later, degraded fast. If you remove the poly A2, the situation is a bit more complex. It's always looking for somewhere where to polyadenylate, and there is some autoregulation, but it's a very artificial situation. It doesn't find out the splicing a poly A site and goes around looking for it. Maybe uses a lot more PA4, but we don't see it in these amplifications. But we don't see it also in northern blots. If you duplicate, you remove the poly A2 and put the poly A1, you get a read-through. So poly A1 is used, but also a little bit poly A2 is used because the pol2 goes through the junction, but the regulation still persists. Obviously, when you have the deletion of the whole area and you have a sort of a cDNA, then obviously there is no regulation. There is no regulation on a single RNA species when you mutate the splice sites, and so on and so forth. This, as a reference, is the endogenous protein that actually makes a nice switch. So this is a situation at the level of RNA. After a lot of confusion, I think that we have it more or less clear. And the question is then, well, there is another question, actually, that is we were asked by reviewers. This area has any importance? Then if you express a gene that ends in 100 nucleotides downstream of poly A2, there is no effect. I mean, you have autoregulation all the same. So at least not significantly, this area does not contribute significantly to the self-regulation process. In collaboration with Nick Proudfoot, early on, we have done BRDU and ChIP-POL2 and SER2 IPs, and the result was that both before and after induction, before is the white boxes, after is the black ones, the bars, and you see before and after there is a higher density of POL2 at the first exon. Then there is a drop in this area, and then there is an increase again in this area, a slowdown, higher density, that increases a lot with TDP-43. Then also an interesting observation is that if you assess the different messengers before and after induction, dividing nucleus and cytoplasm, that poly-I2 that I was showing you seems to be very hard for it to go to the cytoplasm. So essentially, after induction of higher amounts of TDP, you have two events. One is the splicing of intron 7, and the other is a switch from poly-I1 to poly-I2 that have this characteristic, is more retained in the nucleus, and is a very low amount. So to this point, the model was the POL2 goes through, polyadenylation. When there is induction of higher concentration, TDP is always there, so some there is there, so more density in this area anyway. When you have higher levels of TDP, you get more binding in this area, and there is a slowdown or accumulation of POL2, and some of it probably fall out, and there is early termination of transcription. Some of them may go to poly-A4. So you have three probably outcomes: the splicing of this intron and the use of poly-A2. You have degradation of RNA that actually is badly terminated, or some poly-A4 full transcripts that you saw in the northern. This one persists quite a bit. Which of these three situations is the one that is critical for autoregulation? And that's the next question that we ask, and decided to make, the splice sites are reasonable, not a great deal. You have a 3G there in the 5' splice site. There is not a very nice polypyrimidine track in the 3' splice site, so you can shift it to a super site, something that will score 1 in the programs for recognition, and at that point you get a super splice construct that constitutively, without any induction of TDP-43, it doesn't make any difference if there is a lot of TDP-43 or not in the cell, produce a single messenger after a splicing using the poly-A2. One would expect that this messenger is produced, is the 100% of the construct, so it should be quite abundant. Actually, here we compare co-transfecting the construct X7, the super splice construct X7 is the wild type 3' UTR, and both together, and you see that the ratio is quite low of this messenger, and we see the same for the protein. The protein is low when you have a good splicing in that region, very low. It's something that is strange for us, it was. Here you have the comparison of the X7. Here the self-regulation didn't come very well, but still there is quite a bit. Here obviously there is nothing. One important thing, here is the protein, sorry. This is the protein, and we have the protein, the messenger. And we have the PCR, we have the cycloheximide to prove that there was not NMD, and you can see that there is no particular increase in inhibition. We see the same with UPF1. So the mechanism, for some reason, is not NMD when you have this splicing there. And so which is it? So we ask the question, is the poly-A2 intrinsically a bad polyadenylation site and will not be capable of sustaining translation or something like that? So we make a cDNA that instead of ending up in poly-A1, will end in poly-A2. And the transcript will be identical to this one. The difference is that this one is produced by splicing, while the other is just by transcription. Okay? And in that case, we have equivalent amounts of the two messengers and a one-to-one relation to this. Already? Okay. I have discount for the beginning. Yes. Okay. I am finishing. I am finishing. So you see, this poly-A2 is not intrinsically worse. And actually translating, I don't know if I have the slide of the protein, no. And actually translates very well in protein. So no autoregulation, but is perfectly capable of getting out of the nucleus, getting translated, stable, and so on. So this difference between this messenger here and this one, they are the same messenger. It's the way that they were made, the difference. So what it makes the difference? Why? We look at the cellular localization of these messengers, and this is the normal construct, let's say what will be the wild-type 3' UTR, GFP signal from the protein, RNA fish, and you see that most of the messenger is in the cytoplasm. For the cDNA that ends in poly-A2 is the same story, but this messenger is the same than this one, but this is made by splicing, and this one actually is not as abundant, and there is a lot of signal also in the nucleus. You see it more clear here. So the splicing is been marking in some way the messenger is being made, and signaling its degradation is not exported from the nucleus or whatever. So the model that we have essentially is this one. If you have a very good splice site here, you trigger the process of splicing, but as for some reason that we still haven't been able to map at the sequence level or anywhere else, this intron in the 3' UTR is not liked. The spliceosomal complex probably is mostly non-productive, so it will be seen as an intron retention that we were talking before yesterday morning. So intron retention, that is a mechanism for control. But this normally is not an intron, so the way that the cell is handling it is, if a spliceosome forms there, then yes, it's an intron, but it's in the wrong place, should be degraded. I don't come with any better model at the moment. A little bit of poly-A2 is made, but in these conditions is retained in the nucleus and degraded. But if you do the cDNA of this one, you get enough protein and messenger outside. So we still are looking around, but seems that the process, the central question in this process is the spliceosomal formation, a non-productive spliceosomal formation. Just to give a very brief switch to the disease, in the neurons of patients with ALS, you find amyotrophic lateral sclerosis or frontotemporal dementia, you find inclusions of TDP-43. These inclusions, TDP-43 normal as shuttles from nucleus to cytoplasm, and when there are inclusions that we don't know why they start, TDP is trapped in the cytoplasm of those in those inclusions and is being missing from the nucleus, and then its functions in the formation, for example, of the neuromuscular junction are lacking. And you are going to hear a little bit from Emanuele of what we know about the aggregation formation, etc. So if you have any questions, well, okay?